All right, welcome back to episode 7 of the Biochemistry MCAT Prep series. And today, what we're going to be doing is going over 10 practice questions based on the last 3 videos. So let's get right into it. So, the first thing I'm going to do is just give about 5 to 10 seconds at the beginning of each question. If you guys want to pause the video, try and figure out the solution, and then come back and we'll work through a solution together. Right, so question 1 reads, a researcher studies the folding pathway of a newly discovered enzyme. So right off the bat, we're talking about a folding pathway. The unfolded protein exists as many possible conformations. As folding occurs, the number of possible conformations decreases until the protein reaches its functional state. And this makes sense. Remember, if you have a protein that's unfolded, looks something like this, all these arms can bend and rotate in many, many different ways. So that makes sense. So now our picture in our head that we should be thinking about is we're starting with some sort of string, some sort of long peptide, and we're trying to fold it into one functional protein. So let's think about these answers in the context of that idea. So moving on, which statement best explains why the folded protein is favored despite having fewer possible conformations? And so even before we get into the question, one thing that immediately strikes me about this question is we're talking about a folded protein being favored. And so in the back of my head, I'm just going to be thinking about delta G. Because remember, delta G or free energy is what drives protein folding. Okay, so let's look at the answer choices. Answer choice A, the decrease in protein entropy is compensated by a greater increase in solvent entropy. Okay, that seems pretty reasonable. So let's just keep that in the back of our head. Remember, when we talked about the hydrophobic effect, one of the big or main takeaways from that lesson was that even though you're making the interior of a protein more hydrophobic, for example, by putting a bunch of hydrophobic valine groups together, you are compensating for that decrease in entropy because you're making less randomness. You're making more order by putting all the hydrophobic groups together. You are able to compensate for that by increasing the entropy of the water, which now has more ways it can move around the hydrophobic group. So that answer choice seems reasonable. So let's move on. Let's just go through the rest of the answer choices. B. The folded protein has greater entropy because it forms more intramolecular bonds. So one thing that's right off the bat wrong with this answer choice is we're talking about greater entropy because we're forming bonds. So if you're forming intramolecular bonds, you're decreasing the randomness. Think about it. If you're making bonds and forcing molecules and forcing peptides to stay in one particular orientation, you're decreasing the amount of movement, you're decreasing that randomness, and so you're decreasing entropy because entropy can be thought of as randomness. So that answer choice is wrong. Moving on. C. The folded protein is favored because all enthalpic interactions increase entropy. Well, we know that's not the case. Remember, enthalpy is related to delta H, and entropy is related to delta S. It's not true that every enthalpic interaction increases entropy. For example, if you break a bond, you are increasing entropy because now you're allowing the molecules that were formerly bound to each other to have more movement, to be able to move freely. But then on the other hand, if you make a bond, you're now decreasing entropy because you're holding those two molecules together and limiting their movement. And so even though making a bond is an enthalpic interaction, we see that it leads to a decrease in entropy, not an increase. And so answer choice C is wrong. And finally, answer choice D. The folded protein always represents the global minimum energy state regardless of environmental conditions. Well, we know this isn't the case. We know that the native state of a protein depends on the environment. So, for example, we know that changing pH, changing heat, changing what molecules are in solution, all can impact how a protein folds and how it assembles in solution. And so for question 1, that's why the correct answer choice is A. Okay, so moving on to question 2. So question 2 reads, a mutation replaces a valine residue buried within the hydrophobic core of a protein with lysine. So right off the bat, we're replacing a hydrophobic residue with a charged residue. And we're doing this inside the hydrophobic core of a protein. The mutant protein folds poorly compared with the wild type protein. Which explanation best accounts for this observation? A. Lysine causes immediate cleavage of nearby disulfide bonds due to electrostatic repulsion. Well, this doesn't make sense. Remember, disulfide bonds are covalent. And so we're not just going to be breaking covalent bonds due to electrostatic repulsion. That's nowhere near strong enough to break a covalent bond. B. Lysine prevents formation of peptide bonds because it has a positively charged side chain. Well, we know that's wrong. Lysine can be inside of a peptide just like any other amino acid. And so just because lysine has a charged side chain, it doesn't mean it's not going to be able to form peptide bonds with other amino acids. And so that's why B is wrong. C. Lysine eliminates hydrogen bonding between backbone atoms required for secondary structure. Well, let's think about that. Let's pretend we have a serine, a lysine, a serine on one side of our protein, and then a serine, lysine, serine on another side. We know that the serines have OH groups in their as part of their R groups, and we know that as a result, they can bind with each other via hydrogen bonding. So let's think about why the presence of a lysine would inhibit, in this case, either of these two hydrogen bonds being formed. And if you think about it, there's really no good reason, because it doesn't happen. Why would a lysine in between two independent peptides inhibit the hydrogen bonding of those peptides? It wouldn't. So that's why answer choice C is also wrong. And so now by process of elimination, we know that D is the correct choice, but let's just go through it and understand the logic of why that's the case. Lysine disrupts hydrophobic packing because its charged side chain is unfavorable inside the protein. So remember, this whole question is based on why the mutant protein folds poorly compared with the wild type protein. And we know that one of the biggest factors or biggest driving forces for protein folding is hydrophobic interactions. And so what we're doing here is we're replacing a neutral residue, a valine, with a charged residue, a lysine, and we're doing this inside a hydrophobic environment. And so think about it. You have an environment where everything inside likes to be neutral. It likes to be hydrocarbons uncharged. And now you're adding a positive charge into that environment. Well, that positive charge isn't going to be stabilized by anything. It's surrounded by stuff that's completely neutral and can't stabilize it. So energetically, it's going to be very unfavorable for it to be in there. You're taking a lysine, which is charged and wants to be surrounded and stabilized by waters, and putting it in an environment where it can't be stabilized at all. And so that's going to be energetically extremely unfavorable, and that would impact how a protein folds, because that lysine is going to take a lot of energy to be forced into a hydrophobic environment, and often that's just not going to happen. And so that's why answer choice D is the correct choice. Okay, question 3. An enzyme has several acidic amino acid residues involved in salt bridges, stabilizing tertiary structure. Okay, so we're talking about salt bridges and acidic amino acids. That means we're going to have negative charges here, and we're somehow involved in salt bridges. So that means we're having a negative charge interacting with a positive charge. The enzyme loses activity when placed in a solution with a pH of 2. Which mechanism most likely explains the loss of function? Well, let's think about that. We have an amino acid, for example, like aspartate or glutamate, that has a negative charge. Because remember, let's say for aspartate, for example, this is the side chain. And so this OH at physiological pH is going to be a negative charge. And so we're involved with some other amino acid, like for example, a lysine, that's going to end up having a positive charge. And so these two charges will be interacting, and we're saying that this bond is essential for tertiary structure. And so we're taking this situation, which is occurring at a pH of 7, and now we're moving it to a pH of 2. And so what's going to happen? Well, this negative charge on this aspartate is now going to turn back into a proton. We're going to be protonating at a low pH. And so now we have something with no charge and something with a positive charge. Well, these aren't going to form a bond. And so we're not going to be able to stabilize that tertiary structure. So let's look at an answer choice that might reflect this logic. A. Acidic conditions convert alpha helices into beta sheets by altering peptide bonds. Well, that's not what we talked about. B. Increased hydrogen bonding between acidic residues prevents substrate binding. Well, this has nothing to do with substrates. We're talking about stabilizing structure. C. Protonation of acidic residues disrupts ionic interactions, maintaining tertiary structure. And that's exactly what we talked about. Protonation of an acidic residue disrupts ionic interactions and affects tertiary structure. And then D, low pH causes hydrolysis of all peptide bonds within the protein. Well, that's not the case. Remember that pH is going to affect, secondary, tertiary, quaternary structures, but it has no effect on primary structure. Question 4. A mutation causes cells to produce dysfunctional molecular chaperones. Newly synthesized proteins aggregate in the cytoplasm. Which statement best describes the role of chaperone? And so this question is really just testing whether you know what a chaperone does roughly. And so it's not asking you to, for example, know the role of a specific chaperone, just in general, what is the role of these molecules? And remember, in a previous video, we talked about chaperones as being molecules that assist protein folding. And so with this definition in mind, let's go through the answer choices and see which ones make sense. So A, they permanently stabilize proteins by forming covalent bonds with substrates. Well, this doesn't make sense. Chaperones do not form covalent bonds with proteins, and they don't permanently stabilize proteins either. Remember, think about proteins as being made in a ribosome. During that process of being made, remember, chaperones will come along and stabilize the protein as it's being made to help with folding. But then after the protein is produced, after it's done being made on the ribosome, that protein is going to be folded. But those chaperones don't stick around. Remember, their job was just to help with folding. Once the protein is done being produced and folded appropriately, they're not staying there. They are being released back in the solution to help with the folding of more proteins. I mean, think about it from a cell's perspective. If you're investing energy to make chaperones, you'd rather that they're being able to be reused and not just used once to fold one protein. And so that's why answer choice A is wrong. They don't permanently stabilize proteins, they only help stabilize them when they're being folded, and then go stabilize other proteins as they're being made. And they don't form covalent bonds with substrates. Remember, substrates are what you add to proteins, or what proteins interact with to produce a certain function. So this answer choice in general just doesn't really make sense. B, they determine the final amino acid sequence required for proper folding. Well, this doesn't make sense. Remember, we're going DNA, RNA, proteins. And that's really like the central dogma of biology. That's a super classic example. And so nowhere in this sequence is chaperones. And so that's why answer choice B is wrong. D, they provide an environment that reduces inappropriate interactions between unfolded proteins. Okay, that seems reasonable. They're making an environment that enhances stability and enhances protein folding. They're reducing inappropriate interactions. So that seems about right. And then D, they replace the need for hydrophobic interactions during folding. That's not true. If anything, they enhance protein's ability to find appropriate hydrophobic interactions in order to fold correctly. And so that's why answer choice C is the correct choice.